Hi, my name is Ethan, and today I'm going to be talking about SSRIs. SSRI stands for Selective Serotonin Reuptake Inhibitor, and it's one of the most commonly prescribed medications in the world, used to treat depression, anxiety, and OCD. To understand how SSRIs work, we need to go deep into the serotonin signal transduction pathway. Serotonin, chemically known as 5-hydroxytryptamine or 5-HT, is a neurotransmitter, meaning it's a chemical the brain uses to send messages between cells. More specifically, it belongs to a class called monoamines, which are messengers built around a single amine group. Your body actually produces serotonin from tryptophan, an amino acid you get from food, and it's made primarily in a cluster of neurons in the brainstem called raphe nuclei. From there, these neurons project through the brain, helping to regulate mood, sleep, and cognition. When raphe neurons fire, an electrical action potential travels down the axon toward the synapse. At the synapse, the signal triggers vesicles to release their contents. Vesicles are tiny membrane-bound sacs that store serotonin until it's needed, and when they fuse with the cell membrane, they dump serotonin into the synaptic gap. From there, serotonin drifts across and binds to receptors on the receiving neuron. The two most important serotonin receptors for mood regulation are the 5-HT1A and 5-HT2A receptors. Both are what's called G-protein coupled receptors, meaning they work by activating a relay protein called the G protein sitting just inside the cell membrane. The 5-HT1A receptor activates a G protein that inhibits an enzyme called adenyl cyclase, which lowers levels of a signaling molecule called cyclic AMP, generally reducing neuronal activity. The 5-HT2A receptor activates a different G protein that stimulates an enzyme called phospholipase C. That enzyme produces two molecules called IP3 and DAG, which are second messengers, meaning they carry the signal deeper into the cell. They trigger calcium release and activate protein kinase C, a protein that switches other proteins on or off downstream. Together, these cascades shape gene expression, synaptic strength, and long-term emotional regulation. Once serotonin has done its job, a transporter protein called SERT, short for serotonin reuptake transporter, pulls it back into the original neuron. There, it gets either recycled or broken down by an enzyme called monoamine oxidase, which chemically dismantles it so the signal stops. In depression, SERT works too aggressively. Serotonin gets pulled back before it has fully activated its receptors, leaving those downstream cascades chronically understimulated. SSRIs solve this by binding to SERT and blocking it. With reuptake inhibited, serotonin stays in the synapse longer, binding its receptors repeatedly and sustaining both the 5-HT1A and 5-HT2A cascades. Over weeks, that sustained signaling gradually increases production of BDNF, or brain-derived neurotrophic factor, a protein that promotes neuron growth, repair, and resilience. This slow rewiring of neural circuits is the actual antidepressant effect, which is why SSRIs take several weeks to work rather than acting immediately. So SSRIs don't create new serotonin. They protect the signal that's already there and give the brain the time it needs to heal itself. I hope you enjoyed my video and learned something about SSRIs. Thank you for watching.